Hello guys, in this video we will discuss about sample paper set 7 section 4 question number 1 to 5. Let's start. Here the first question is show the short answer type question. The first question is define a chemical equation and how do you balance a chemical equation. So the chemical equation is a balanced equation is an equation of a chemical reaction in which the atom of an element in the reaction and the total charge is the same for both the reactant and the product. It means it is a reaction which are balanced, which is the number of reactant and the number of product is equal. So there is a reaction is called balanced reaction. So there is a step to balance the reaction. There are more type of reactions like acidic reaction, basic reactions, neutralization reaction, redox reactions. There are many type of reactions. So we need to balance and we need to equal the atom and the hydrogens. So there are two methods to balance the equation. The one is hydrogen method, second is basic method. So there is a steps, there is a 5 steps to balance the chemical equation. First is identify the most complex substance. Second is beginning with that substance, choose an element that appears in a one reactant and one product. Third is balance polyatomic ions as a unit. So fourth is balance the remaining atoms like hydrogen, H2O and oxygens extra. Fifth is count the number of the atom of each kind on the both side of the equation. Be sure that the chemical reaction is balanced. So there is a steps. I give you example. This is an example is combustion of heptane. Heptane means 6,7S16. There is a combustion means addition of O2 gives CO2 plus H2O. There is a reaction we need to balance. Firstly, we need to balance the carbon. First 7 carbon and their left side is right side is 1 carbon. So balance 7 for right side. For 7 carbon is okay. Then balance the hydrogens. Then there is a 16 hydrogen and then there is a 2 hydrogen. So balance 8 hydrogen in right side. So and at last oxygen. Oxygen of left side is 2 and right side is 2 plus 8. 2 plus 8 10. So there is a 2 is left side and 10 is right side. So left side 11 with balance. So total is 12. So there is a reaction of balance 22 oxygen total on the reactant and the product side. So C7H16 plus 11 O2 gives 7 CO2 plus 8 H2O. So there is an answer. Next question is give at least 3 function of small and large intestine. So the answer is basically intestine are used to supply the food into the body. There is a pipelines like small pipelines and large pipelines. So the small intestine basically break down the food more thoroughly than the part of the digestive system. For example, it breaks down the sucrose into the glucose and fructose. There is a 2 isomers of the sucrose and fructose. Through saccharase, pentose in amino acids. Amino acids are also important in our body. Through acidic and fatty acids also. So the glycol that have lips. These are only some of the digestive process that take the place in the small intestine. And the large intestine is the other words, they do not conduct for the digestion. It does not participate in the directly to the digestion. It is absorb the water and the mineral salts from the undigested food into the bloodstream before the digestive ancestor food proceed to the rectum. So there is a basic work. Next, what is placenta and mention its role? Basically placenta is like a disc like tissue. It's a tissue. It's a or look like a disc from by uterus wall or as some anaerobic part. Placenta is there for a special tissue of the 2 individuals. It is made of the finger-like projection or villi of the embryonal tissue, which are present on the blood-filled spaces. If mother tissue like intestinal villi provide a large surface area for exchange of material between the mother's blood and the embryo. So the function is there is a lot of functions. The first function is through placenta the developing embryo gets oxygen and nutrients like glucose. It provides the oxygen and the nutrients to the body. So waste generated. Second is a waste generated by the umbilical or special to mother blood through like placenta. And the third is the embryo gets some new power against disease from the mother dish as placenta. Placenta is basically the supply the foods against the disease, cure against the disease from the mother. Next is describe the Mendel dehybrid cause in the pea plant in a punny checker board. It is a main question for biology class 10th. So there is a breed combination YYRR on the right side diagram, YY small rr small I YRR. So there is a breed table. So learn this table. So the basically a breed table scores the experiment in the genetic in the phototype of a two generation follow through the mating or individually carefully multiple analysis at those genealogies. Basically it's a combination. Scientists made two more combinations to take a breed and what to produce, what to hybrid the different peas and different structures, different copies and two types of each genesis. So basically mostly sexually reproducing organisms are carrying two copies of genes, allowing to carry out different alleles. Historically, organisms from two different breeds lines as a hybrid. So they can hybrid and then take different hybrids. So true breeds is generally as a homozygotes. So there is a gametes YYRR, small capital RR, small y. So there is a ratio. He found seeds for 4 types in 9:3:3:1 ratio. There is a hybrid ratio. So there is an answer. So the next and last question is, what is the difference between series and parallel resistor? It is a physics question, very important question. So the series resistor, what is the work of series resistor? Series resistor means it's a voltage current is flow in and resistance flow in the series combination, not one by one, not in a one by one. It's a continuous series, like 1 then 2 then 3 then 4. It's a shown in a diagram like V1. What is V1 plus V2 plus V3? Series current is I is equal to I. Current is not additive. Remember this, current is not additive. So I1 is equal to I2 is equal to I3. But resistance and voltage is additive, R1 plus R2 plus R3. So basically series resistor is the same amount of the current flow through the all components. It's a current is through the all component in a one time. In the electric current component are arranged in the serial lines. So there is a line series. So if a component break down, the whole circuit is burned out. If the one circuit is break down, then the whole circuit is empty. So there is a disadvantage as a. So the parallel resistor means it goes one by one, like one then 2 then 3 then 4 in the row. So there is a V1 is not there in this voltage is not additive is equal to V1 is equal to V2 is equal to V3, but current is additive. I1 is equal to I2 is equal to I3, but the resistance is reciprocal. Reciprocal means 1 by R is equal to 1 by R1 plus 1 plus 1 by R2 plus 1 by R3. So there is this is parallel combination. So the parallel combination, the current flowing through each component combines to form the current flow through the source. In the electric circuit, components are arranged parallel with each other. When the other component will function, every one component breakdown, each has their own independent circuit. Means if first circuit is breakdown, then second and third is individually, they works continuously. So there is a benefit and there is an advantage. So the parallel is good for then series, there is a advantage.